Thank you very much for the introduction. It's a great honor to be here, to be able to be involved in the inauguration of the Hillman Laboratories in Singapore. I was very much involved with Hillman for a number of years, and very reassured by the welcome we're getting and also the facilities I see here in Singapore. So what I'm going to do is I'm actually going to give a presentation that I've never given before, really. It's really about a new direction of travel for Wellcome. I'm currently the director of the Wellcome Infection Health Challenge. So for those of you who don't know what the Wellcome Trust is, it's been established actually, it's a philanthropic organization which was built upon a pharmaceutical company called the Wellcome Foundation. And our current endowment has actually just increased dramatically. It's now at around £38 billion. And this is our chair, Gillian Gilead, and our director, Jeremy Farrar. And what we do is we invest in research and development. And we estimate that we'll probably spend around £16 billion over the next decade in terms of a targeted investment, and I'll try and describe how we're going to go about doing that. But we have the potential to also make larger blocks of investment. If you want to do something, for example, we established the Wellcome Trust Sanger Institute to challenge the Human Genome Project and to deliver the sequence of the human genome. We can take initiatives at scale, or we can take funding in terms of diversity. But we're not for profit. So what we've actually recently done is we've undergone a reorganization. So historically, Wellcome has been underpinned by discovery science. It's different from Gates and many other organizations in that we're built on science, and that science is discovery science. And that science will stay. We fund mainly that in the UK, but also in low and middle income countries. And we can support places like the Laboratory of Molecular Biology in Cambridge, the Crick Institute, the Wellcome Sanger Institute, overseas units. So it's very diverse in the kind of science that we support. But as soon as we're entering the translation area, we've decided to focus on three very discrete areas. Area one is mental health, which I won't talk too much about, and this is run by Miranda Walpole, a colleague of mine. We've just entered the field of climate health for the first time. We've just recruited in Alan Dangor, who is our new head of climate health. And I'm currently looking after the infection challenge. So historically, Wellcome has been associated with infection research, but it's always been very spread around the organization. It's never been in one area, one umbrella. And so what we've decided to do is to bring all of our infection work together and to manage it more strategically in the translational area, and also to connect it very strongly to our R&D portfolio, our research portfolio. So the research portfolio, the discovery portfolio can then drive our scientific innovation in infection. And around that, we have things like data health science. We're very strong on bioinformatics historically because of the Human Genome Project. And we integrate all of this we meet regularly and try to integrate ourselves into a common strategy. So our philosophy in the infection area we've been working on is really around this definition. So we want to use science and policy to stop diseases from escalating. We're not so much worried about the current disease burden. We're much more interested in preventing the diseases in the future and trying to limit the disease burden by turning off the drivers and sources of infections. We want to do that by looking at organisms which are likely to be new pathogens like COVID, but also looking at organisms like AMR agents that are escaping therapies or vaccine evasion. The second thing is we want to concentrate our efforts globally in this area, so we're not restricted to the UK, and we want to work more closely with those communities that are the most disadvantaged. So we will be investing a lot more money south compared to north. Our discovery will stay predominantly in the north, but our translational will move much more towards the south. And so this is really a guiding principle of how we're going to do our future investments. And we've got three main drivers here. One is to gain a greater understanding of the mechanisms underlying the sources and drivers of infectious diseases, or what selects the next generation of threats. And we'll do that through a scientific approach. And we'll do it by hypothesis-driven research and also by using data and collecting data. The second thing is we want to try to implement, we want then to try to turn the paradigm from treating the disease burden much more to a focus on intervention and prevention, and try and get that pendulum moving towards much more about the prevention and intervening and preventing outbreaks rather, and also identifying outbreaks earlier on and intervening earlier on. And the third area is really about the idea of creating a new generation of affordable products. So that involves starting at the very beginning, right in discovery, and moving right through to impact, and trying to look at all of those different stages and identifying mechanisms where we can bring not only affordability, but also accessibility to make sure that if we make an affordable product, whether it's an antibody, a vaccine, a diagnostic, or some kind of intervention, that it has impact. So we've organized ourselves. I'm currently the director, but I laid this out, the challenge area around areas of expertise. So if you look across the top here, we have our heads, and one area is epidemics and epidemiology, which underpins everything we do, maybe potentially surveillance and interventions. The second area is prevention, where we've got Charlie Weller, who's looking after vaccines, human investigations. We do a lot of work on human challenge, and antibodies and immunity. And so disease tracking and reservoir sits in epidemiology, and that works very closely with our early interventions. Interventions is very much around therapeutics. We've got a huge therapeutics portfolio, and we've brought alongside therapeutics drug resistance know-how. So the idea is for the drug resistance, the AMR experts to work very closely with those developing therapeutics, and then to link that into diagnostics. And finally, we have a fairly new area, which is really around systems biology, maybe modeling infections, the sites and systems. So if we're going to be effective internationally, we have to develop sites. We're interested in Singapore, for example, as a site. What can we add value to by bringing things like the Hillman together with other entities and connecting into regions about how it, not only in terms of interpersonal interconnections, but also we're very interested in data management, data connections. And finally, the other area which is really interesting is climate and infection. We started the first projects really on looking at how climate will influence infection. The first project we've funded is the idea of the data collection for infection is completely separate from the data collection for climate. So the first things we're going to do is to bring in data management systems. We're asking for funding in that area to actually bridge that gap between bringing data know-how in climate together with data know-how in infection. That's how we're going to build our platforms. We're interested in the effect of temperature on infection as well. So we've got a whole repertoire. If you look across this list of, if you like, initiatives that we've supported either previously or we're interested in supporting, it's quite an impressive reach that we have. We've talked about CEPI already. CARB-X is our initiative on antibiotic development, the Hillman Laboratories. We run and fund the World Mosquito Program, which if you'll be familiar with, a dengue challenge area. But there's a whole array of different areas that we actually focus. And all of those now fall under a single umbrella with the infection challenge area. Previously, they were all separated. But now the idea is we will prioritize and we will manage those as a portfolio. They will not be operating independently. For example, we can connect the World Mosquito Program to our climate health area. And that's the first part of the work that we're going to do in climate. We're going to look at the influence of climate on vector biology, on the transmission of diseases. One of our assets is the Wellcome Sanger Institute. I was head of pathogens there for 10 years. And we'll start to align the Wellcome Sanger Institute with our strategic aims. You know, we have this huge resource, really, that has driven, for example, it's probably not well known that 25% of all released bacterial genomes, the 20 most common pathogens, have been generated from the Wellcome Trust Sanger Institute. 95% of all Anopheles mosquito genomes have been generated by the Sanger Institute. And 90% of all malaria genomes has been generated from the institute. And this was built really around our know-how around human genetics. We put on top of that the capability to operate at scale, and that we're going to connect that to our global networking. So we're going to retune this powerful asset and align it more initially with infection, but potentially into climate and potentially into mental health. We were able to act upon this know-how, 20 years of experience in the pathogenomics area. I attended a meeting about 2 years ago, about 2 weeks after the arrival of COVID into the UK, and we formed this consortium called COG UK, which I was in charge of the governance structure of COG UK, with the aim, driven by Patrick Vallance, to set up a sequencing entity for COVID within the UK. And interestingly, the biggest challenge we had was to connect sequence to human data. There was no mechanism where you could actually take a sequence and connect it to a data from a patient in this realm. And the people who solved that problem, ironically for us, were the human geneticists who have been doing this for a long time and dealing with privacy. But we were able to catapult off this platform, headed by Sharon Peacock here. And we also had this huge array of different open access data tools. Pangolin, for example, is the tool that was used to identify the first variants of COVID. It was this consortium that identified the COVID variants using this armory of biotechnology linked to this generation of information platform. And we want to extend this into other parts of the biological world, and also huge resources in terms of compute, which are managed ethically, open access, and securely. So we're actually building upon this. And for example, a year ago, I started funding COVID sequencing built upon COG UK. We have a process called COG Train, which actually trains people in LMICs to manage not only the data, but also the sequencing, and also we're going to build upon that to actually go into phenotyping. But because we had this 20-year legacy of training individuals in pathogenomics, we trained fellows and investigators right across the globe. Within a few weeks, I was able to, just by phoning around, to identify people across the globe to get involved in the consortium. For example, we had an investigator in Buenos Aires who was connected to PAHO, and immediately we were able to get coverage right across Central and Latin America, COVID sequencing, and we've invested in other parts of the world as well, particularly into parts of Africa. But this is quite a useful system for us, because although it's a network for working on COVID, we're going to use this network now not only to work on COVID, but to gradually extend it into other disease areas. We'll use that as an international network for data management and processing through our trained cohorts to work on AMR, to work on other organisms and other pathogens, including malaria, etc., and extend that network and consolidate. And again, we're very interested in hubs. You know, there are certain hubs around the world where we can consolidate to nucleate our compute ability, and then use that as a way of connecting the threads that will then get regional connectivity to actually then increase our ability in terms of surveillance. We will not fund the surveillance itself, but what we'll do is fund innovation in surveillance to actually improve our ability to capture data and to analyze it in an open way. And the sort of reach we've got is actually extraordinary. For example, in the Philippines, we've been running pathogen sequencing for cholera and for typhoid over the last 10 years, and we were immediately able to turn that around to COVID. And we cover, for example, through IVI and through Jerome's group, we were in Madagascar, where we've been doing sequencing of typhi, Salmonella typhi, and then again that has also been used by IVI to build upon for their COVID work. So it's actually a very important sentinel system that we've established and sustained. One of the challenges is going to be about how do you sustain such an entity over a long period of time. Governments are going to be challenged with this task. And we've been thinking about this as a problem, and it will be one of our highest priorities over the next few years. I mean, the other area we have to think about is affordable medicines, and it's not just vaccines, we're also thinking about antibodies, we're thinking about chemical entities of small molecules. There are more challenges in, for example, in antibiotic movement through the regulatory pathway than there are probably through vaccines. And so we're going to sit all of our teams together so the people working on antibiotics or antibodies on vaccines sit together and we talk about the barriers to actually getting these materials in an affordable way through the system. And so what we're going to do, we have this fantastic interface, really, between our discovery and our translation. So we'll be focusing on the idea of bringing innovations from discovery into the clinic. That'll be one of our priorities. And we've got organizations like NextX, which is the next version of CARB-X, CEPI. HIT-NTD is our conglomerate of working on small molecules for neglected tropical diseases, and Human Challenge. And what we want to try to do is to change that landscape and make it more efficient and more affordable by pulling things through, particularly in the regulatory environment. We're going to focus on the regulatory environment as a mechanism for actually speeding and improving the movement of affordable medicines through the system. I mean, we've probably got the biggest vaccine portfolio in the world. If you actually look at what we have on our books, for example, CEPI, we work very closely with CEPI. CEPI headquarters are in the same building as, we're on the same floor as our vaccine group. And so we have a tremendous dialogue and influence about how we work together with organizations like CEPI, who work on the epidemic and outbreak diseases that are outlined here. But we also have another portfolio. For example, we have a list over here of vaccines that work on from innovations. Some of them you'll recognize it because some of you are funded in some of these vaccines. GHIT, we fund GHIT, which is in Japan, where the Japanese industry is driving forward vaccines. A lot of them are malaria, you can see some of the diseases. We have our Hillman portfolio, which I don't have on this slide, but we also have a CARB-X portfolio. These have previously been completely separate. We brought them all under one umbrella, and we're going to manage them as a portfolio. This is really important because what we need to be able to do is to identify the areas where not only ourselves but also our partners, we can work together to be more effective. I think if we'd be more effective as a group and as a single entity in bringing some of these very challenging diseases through to fruition, to not only to get the vaccines licensed, but also get them to impact. So that our portfolio is actually available to us for the very first time. The other thing is we want to have an impact. So what we're looking at is innovative ways of actually breaking down some of the barriers towards the generation of affordable molecules. One area we're very interested in is Human Challenge. I'll talk about that, but there are other areas as well. So we've been the promoter of the idea of Human Challenge to actually break the regulatory cycle. Andy Pollard will be talking later on about Typhoid Challenge, which broke really the WHO pre-qualification of typhoid vaccines. It was funded initially, a grant myself and Andy put in to get the Human Challenge going. There's huge resistance in the community, but Wellcome was brave enough to fund it. We've also been funding all of the SARS-CoV-2 challenges, which is generating incredibly interesting data. I've seen a lot of the data. We do that by paying, for example, GMP. We're just doing a GMP Delta variant of COVID, ready for challenge, and supporting that kind of infrastructure. But we're also trying to move that infrastructure from the north into the south. So can we actually then start to use pathogen challenge, not only in the north, but also in the areas where diseases are endemic? But there's something very special about Human Challenge. Human Challenge itself is a means to an end, but what it brings is the very highest level of ethical management of clinical investigations, human investigations, into the pipeline. And so we're going to use that as a way of driving forward improved human investigation studies, and we're going to link that up to our stem cell biology, because we believe stem cells are the way in which we can get more closer to human personalized assays for correlates of protection that can then be used to help and facilitate driving forward better medicines in the future. That's the way we're trying to innovate and try to bring different areas together. This is our, just alluding to our therapeutics portfolio. I won't go through all of our portfolios, but HIT-NTD brings a lot of these small molecules. And you look at some of the diseases that we've targeted and with the different phases we do support centers of excellence. We have a center of excellence in Dundee. Not only does it develop small molecules, but it's got this brilliant group that once you've got a small molecule, it's able to identify the target of that small molecule and the mechanism of action of that small molecule. And we're linking that really to better ways of screening, more intelligent ways of screening. And I'm very interested in developing this area. I'd quite like to develop a new center where we bring together chemistry to make better antibiotics using better molecular microbiology and bring our epidemiology groups right up alongside the molecular microbiology groups, right up alongside really innovative chemistry that thinks about how to get molecules into bacterial cells, where most chemists think about getting molecules into human cells. So there's plenty of scope, really, for working on this, because we do now have our portfolio completely visible. And what we're going to have to do is using our escalating criteria to identify which of those diseases we want to focus on. So we will be having choices. We're not just going to randomly fund any infectious disease. We're going to be very selective about the way we go about trying to support the community in the most effective way to bring escalating events and minimize them. And final slide is I talked about the idea of bringing together different assets that we have. So in Singapore, let's finish on Singapore. We've just brought here part of the Hilleman laboratories. We still have the Hilleman laboratories in India. We're working with IAVI to consolidate that potentially into a new center. And we want to then use that as a way of nucleating the way we invest into that part of the world. But we also have funded a clinical trials center in Singapore at the National University here, run by David Patterson. And the idea there is to innovate in clinical trials. We believe this is one of the biggest barriers to the development of molecules. For example, I've just been quoted a price of around $1 billion for a phase 3 study. And I believe we can break that down by being much more innovative, but also talking to regulators in scientific and sensible and ethical ways. I've spent the last 2 years actually in the UK on the MHRA panel for regulating COVID vaccines. And it's been very interesting to see how the regulator not only is willing to move, but also the way that the regulator is willing to listen to good science. And so I think that is an area which is going to be rich in opportunity. And this center within Singapore, I would like to align a little bit more closely with Hilleman and to think a little bit about not only in Singapore, but how might that connect to a center in Melbourne, or how might that connect to a center in Bangkok, or how might it connect to then a center on the African continent. And I've finished, I think I'm just about running out of time. So what I've finished is really, we also are interested in other areas. We're interested in antibodies. We've just done a big review, really, of affordability of antibodies. We're interested in whether or not we can make antibodies affordable. And I'll finish there. Thank you very much.